Hi everyone, welcome to MR Learning Circle. I hope everyone is fit and fine and doing good in their lives. So this is the 5th lecture in our MCQ series for covering the microbiology section of your upcoming GATE examination. So in this lecture we are going to cover methods in microbiology part 3, and the topics for the lecture is PCR and next generation sequencing technology. So let's start. And I hope you all have watched the previous videos, all the 4 lectures, and you can find the playlist. I will pin it here, and if you have not still seen the videos, then please go and watch it. That would be very helpful for your upcoming examination. So let's start with PCR. So the first question is, the fundamental role of tagged DNA polymerase in PCR is: option A, denaturation of DNA at high temperature, B, primer annealing, C, extension of DNA strands at high temperature, D, ligation of DNA fragments. So the correct answer for this question is option C, that is extension of DNA strands at high temperature. So what is the role of Taq polymerase? Taq polymerase is a heat stable enzyme. And it helps in the synthesis of new DNA strands by adding nucleotides to the primer during the extension step. Now question number 2. In PCR, the primary function of primer is A, to provide a starting point for DNA synthesis, B, to separate DNA strand, C, to stabilize Taq polymerase, D, to act as fluorescent probe. So the correct answer for this question is option A, to provide a starting point for DNA synthesis. So primer, what are primers? They are short oligonucleotides. They are complementary to the target DNA sequence, and it provides the 3-OH group for the initiation of the polymerase. Now next question. Question 3. Which of the following modification in PCR can improve specificity when non-specific amplification occurs? Options are A, increasing template concentration, B, using a high annealing temperature, C, reducing primer length to less than 10 base pair, and D, increasing extension time. So the correct answer for this question is option B, using a higher annealing temperature. Question number 4. Nested PCR is primarily used to A, quantify DNA molecule, B, reduce contamination, C, increase specificity and sensitivity of amplification, D, sequence the PCR product directly. So the correct answer for this question is option C, increase specificity and sensitivity of amplification. So this nested PCR, it uses 2 sets of primer in a successive reaction. The second set of primer targets within the first PCR product to reduce the non-specific amplification. Okay, now next question is question 5. The principle of PCR is A, selective amplification of a specific DNA segment using repeated cycles of denaturation, primer annealing and extension. B, ligation of DNA fragments to form recombinant molecules. C, sequencing DNA using chain termination. D, transcription of RNA into complementary DNA. So the correct answer for this question is option A, that is selective amplification at a specific DNA segment using repeated cycle of denaturation, primer annealing and extension. Now question 6. In real-time PCR, quantification of microbial DNA is based on A, measuring the intensity of DNA bands on agarose gel, B, monitoring fluorescence signal proportional to DNA amplification during each cycle, C, counting colony forming units after PCR, D, measuring optical density of bacterial culture. Correct answer for this question is option B, monitoring fluorescence signal proportional to DNA application during each cycle. Now Question 7. The threshold cycle, that is CT, in qPCR, that is quantitative PCR, represents A cycle, the first cycle of PCR, B, the cycle number at which fluorescence exceeds background and become detectable, C, last cycle of PCR, and D, total number of cycles needed to amplify DNA. The correct answer for this question is option B, the cycle number at which fluorescence exceeds background and becomes detectable. So CT is inversely proportional to the initial amount of target DNA, and lower the CT indicates higher starting template concentration. Simple funda. Question 8. SYBR green dye in real-time PCR binds A, only single-stranded DNA, B, only RNA molecules, C, double-stranded DNA, emitting fluorescence proportional to amplification, D, specific primer. So the correct answer for this question is option C, double-stranded DNA emitting fluorescence proportional to amplification. So SYBR green intercalates into double-stranded DNA and fluoresces, and this allows monitoring of PCR product accumulation. Now question 9. Which of the following is an advantage of probe-based qPCR over SYBR green? So your options are A, detects any double-stranded DNA, B, higher specificity due to sequence-specific hybridization of fluorescent probes, C, cheaper and simpler, D, does not require primer. So the correct answer for this question is option B, higher specificity due to sequence-specific hybridization of fluorescent probe. Okay, easy one. Now question 10. Multiplex real-time PCR allows A, amplification of a single gene at a time, B, simultaneous detection and quantification of multiple microbial targets in the same reaction, C, quantitation only after gel electrophoresis, D, detection of only RNA molecule. So the correct answer for this question is option B, simultaneous detection and quantification of multiple microbial targets in same reaction. So that was for your PCR. Now let's move towards the next generation sequencing technologies in microbiology. So first question for this is, the main advantage of NSG over Sanger sequencing is A, lower error rate, B, ability to sequence millions of DNA fragments simultaneously, C, requires radioactive labeling, D, no need for PCR amplification. So the correct answer is option B, ability to sequence millions of DNA fragments simultaneously. Question 2. Illumina sequencing technology relies on A, pyrophosphate detection, B, sequencing by synthesis using reversible dye-labeled nucleotide, C, chain termination with di-deoxynucleotide, D, single molecule nanopore detection. So the correct answer for this question is sequencing by synthesis using reversible dye-labeled nucleotide. Now question number 3. In 454 pyrosequencing, nucleotide incorporation is detected by option A, fluorescent emission, B, release of pyrophosphate leading to light generation, C, gel electrophoresis, D, radioactive decay. So the correct answer for this question is option B, release of pyrophosphate leading to light generation. So incorporation of nucleotides, they release pyrophosphate, which triggering a luciferase reaction, and this reaction emits detectable light. Now next question, question number 4. Which NGS platform allows sequencing of single DNA molecule without PCR amplification? So your options are Illumina, Ion Torrent, PacBio SMRT and Oxford Nanopore. D is 454 pyrosequencing. So the correct answer for this question is option C, that is PacBio SMRT and Oxford Nanosphere. Question number 5. Which NGS method is most suitable for rapid microbial genome assembly and detection of structural variants? A. Short read Illumina sequencing. B. Long read sequencing, that is PacBio or Oxford Nanopore. C. Sanger sequencing. D. Pyro sequencing. So the correct answer for this question is option B, long read sequencing. So long read sequencing can span repetitive regions and structural variants. So this improves genome assembly accuracy. Now question number 6. Metagenomic sequencing in microbiology is used to A. Sequence a single microbial genome. B. Analyze the collective genomes of all microbes in an environmental or clinical sample. C. Only quantify RNA expression. D. Detect only culturable bacteria. So the correct answer for this question is option B. Analyze the collective genomes of all microbes in an environment or clinical sample. Now question number 7. In NGS library preparation, adapters are added to A. Enable PCR amplification and attachment to the sequencing flow cell. B. Label DNA with radioactive isotopes. C. Increase GC count. D. Directly detect fluorescent signals. The correct answer for this question is option A. Enable PCR amplification and attachment to the sequencing flow cell. So here the adapter, it allows fragment to bind to the flow cell and serve as a primer site during amplification and sequencing. Now question 8. Primary limitation of short read sequencing technology, example Illumina, in microbiology is A. High error rates. B. Inability to resolve long repetitive regions or structural variant. C. Slow throughput. D. High cost per base. So question number 8's answer is option B, inability to resolve long repetitive regions or structural variants. So short reads, they cannot span long repeats, making assembly of complex genome challenging. Now question number 9. Oxford Nanopore sequencing detects DNA sequence by A. Measuring hydrogen ion release. B. Monitoring changes in ionic current as DNA passes through nanopore. C. Incorporation of fluorescent nucleotide. D. Gel mobility shift. So the correct answer for this question is option B. Okay. Next is question number 10. NGS based transcriptomics in microbial studies is used to A. Identify DNA mutation. B. Quantify gene expression and analyze RNA profiles. C. Detect protein-protein interaction. D. Measure microbial colony morphology. So the correct answer for this question is option B. Quantify gene expression and analyze RNA profile. So RNA sequencing via NGS, it allows comprehensive analysis of microbial transcriptomes under different conditions. So that was all for today, and I hope you have learned something from this video. And if you have learned, then please like the video, comment down below, and if you have any doubts, please mention it in the comment section, and please share it with your friends and anyone who is preparing for GATE examination. So this video would be beneficial for them. Thank you for watching. Keep studying.